I'm Rich Caruana again from Microsoft Research. This is the second presentation in our session on interpretability for biological systems. Happy to introduce Jack Gallant. Jack is from here at Berkeley, where he's the Chancellor's Professor in the Department of Psychology, but he's also got affiliations in like every other department in the university: electrical engineering, computer science, neuroscience, bioengineering, vision science, and biophysics. Jack's research is in cognitive and human neuroimaging, and he's one of the main people in neuroscience who's focused on what you might think of as naturalistic experiments that are aimed at producing quantitative behavior and models that are really accurate and generalize well. Jack is very proud of the fact that his lab really sweats the details when it comes to data science, and that they use an empirical workflow that's actually quite different from the standard approaches that are used elsewhere in neuroscience. So go ahead, take it away, Jack. Thank you.
I just want to, let's see, first of all, I'm a neuroscientist. You know, I do a lot of applied math, but that's kind of forced. It's not my desire. I just really want to do neuroscience. But because our data sets tend to be more complicated than most people's in my field, we've paid very close attention to the developments in data science over the last 20 years. And so I would say I have glancing familiarity and sort of empirical experience with a lot of these methods. I did want to mention that, you know, the last talk we just saw was awesome. It was an amazing display of discovery science driven by enormous, infinitely large data sets and great machine learning. And this talk's going to be the exact opposite of that. So this is going to be depressing in some ways.
That means it won't be a great talk?
That too. Wasn't the dimension I was thinking of, but that may be also true. Okay, so I just want to mention, because we were just yesterday, there was a lot of kind of philosophical discussions about data science. I just wanted to mention, you know, my perspective on this is the deal here is that the incentive structure for scientists is very different than the incentive structure for engineers. In some sense, you know, we all are doing the same thing. We want to measure some stuff. We want to build a model of the stuff whose dimensionality is far lower than the measurements, right? Because then we can understand it. And then we want to predict some new stuff. Now, if you're a scientist, this is the most important thing, modeling the stuff with a low dimensional model. And in fact, it's so important in, I would say, less developed areas of science like psychology and biology and neuroscience that people don't even worry about this. There's no journal in psychology or biology that I am aware of that requires you to report predictions or effect size or generalization tests, or even to have separate fit and test data sets. So everybody cares about this, and the mark is statistical significance, even though that's the noise floor, not the noise ceiling. And everybody reports significance, and then they declare victory and pretend it's the noise ceiling and move on. So that's science. Engineering, I think to its credit, has to worry about this whole thing, because ultimately, if you have a statistically significant bridge and it falls down, you have a problem and someone will sue you. So the lawyers keep the engineers honest, and at some point, they have to be able to predict. Now, every engineering professor in this room knows that ideally, you want this. You want a model that you can prove. I want to know the bounds, when it'll work, when it won't work, and then I want to show that, predict something, right? But of course, the kids think this is boring. Anybody under 25 wants to do this because it's so much cooler and you can get a job at Facebook. So they want to build neural networks. Engineering professors hate this because you can't prove anything with this. When does it work? When does it not work? I don't know. So this is kind of a stopgap. But if you don't know what to do here, like you got a nonlinear dynamical system or something else that's mathematically intractable, then this is the best you can do. And I think, this morning, as I said, the talk that we just saw was a great demonstration that this is not a pointless exercise. But these two points of view are very different. Engineering professors are willing to do this because they've got to get here first. They've got to get to predictions. And they're willing to take a lousy model first to get to predictions and then go back and try to figure it out. And in neuroscience, this kind of deep network model is kind of pointless unless you can interpret it very, very quickly. So just wanted to mention, it's a kind of a very different thing, very different approach. All right, so I study the brain. The brain is, complicated. That's the first law of neuroscience. There's 80 billion neurons that are organized in these very complicated hierarchical parallel networks. They're organized at multiple scales. The scales don't necessarily communicate with one another. They're kind of compartmentalized. The brain connections are many to many and recurrent. The information that's flowing through the brain networks is very, very dynamic and changes based on the task and over time with learning. And the brain learns continuously at all time scales. So, the brain is very different than the deep networks we use in computer science. Those deep networks, for the most part, are kind of, I would say, circa 1945 neuroscience. And neuroscience, although it's very hard, we've learned a lot since that time. And most of that has not been incorporated into deep networks. But the real problem for a neuroscientist trying to understand the brain is this. We are data limited. At any point in time, in any field of science, you're either model limited, I would say theory limited, or you're data limited. Neuroscience is data limited. It's always been data limited. It'll probably be data limited for a long time. And if you want to think of, if you don't know anything about neuroscience, but you know perhaps about physics, the way to think about neuroscience is it's like astrophysics with a really bad telescope. So you all know the problems with astrophysics. There's billions of stars out there. You can see a few thousand maybe. It's the same problem in neuroscience. And in fact, I've had tons of physicists come through my lab. The only ones that stick around are the astrophysicists, because they just look at the data. They're like, oh, this is the same kind of lousy data I always deal with. No problem. So the brain on a very short time scale of, say, a half an hour is a fixed network. The hardware doesn't change, but the information flowing through the network changes based on attention and learning and the task demands through these control signals that are broadly distributed and no one really understands. So the representation of information in the brain is constantly dynamically evolving. In the human brain, we don't really know exactly what the hardware is. We don't know all of the brain areas. There's probably something like 500 at least different brain structures that all represent different kinds of information in this deep network. And we don't have a very good idea of how these networks represent information. So you can imagine if you take a complicated task like driving, while you're driving through the world, there are constantly changing patterns of brain activity. So you saw when I first put this slide up that we put some slices in the brain and kind of flattened it out. So now we have something about the size of a large pizza. And the red regions are locations where metabolic activity is higher than average. And the blue regions are locations where metabolic activity is lower than average. We're measuring this with an MRI machine. So you can see that these patterns of red and blue are constantly changing dynamically as an individual is driving through this virtual town in this taxi driver task.
One more time, the red versus blue.
Sure. Red here is more metabolic activity and blue is less metabolic activity. This is what the person saw in, so this is an MRI experiment and the person's lying in the MRI machine and they're driving in this virtual world.
And the brain is really the brain of the person watching.
And this is the brain.
Oh, wow.
Yeah, yeah. This is real time. And
the image, or are they turning to like a controller?
I took that slide out. This is an awesome experiment. They're actually driving. They've got a little steering wheel in the MRI machine.
Oh, your video game.
They've got pedals and they've got the virtual reality in a very complicated jury rig system on their eyes. And this is a very large town. It's about 4 square miles on this side. It's got about 200 structures and roads. They spend about 10 hours learning the town. And then after they've learned the town, we just put them in an Uber driver task. Drive to the Taco Bell. And they drive to the Taco Bell. Now drive to the gas station. That's all they do. We do that for hours and we record brain activity. Okay. And then the question is, well, so think about what you do when you're doing this task. Okay, you got to look at the visual input, right? So that has to be represented somewhere. You got to operate the pedals in the steering wheel. That stuff has to be represented somewhere. You have to know where you're going. You have to have some memory of where you've been, where you're going, what the map is. And if you notice in this road, there's other cars and vehicles and things. You have to avoid the other cars. You can't run into them. Can't run into the pedestrians. There's traffic signals. There's all kinds of complicated things going on. All that stuff, the past, the future, perception, motor cognition, all of it has to be represented somewhere in this, right? And we want to know what and where. Right. So, so how do you solve this problem?
Do you also record where the muscle movements are?
The muscle movements?
Yeah.
Yeah. We have all the ground truth from this. So this is a video game, so we can extract all the semantic information from the video game. We know where all the buildings are. We know where the cars are. We know the layout of the world. We we track your eyes so we know where you're looking. And we're recording all the motor movements so we know what your behavior is. So we have all the stimulus task, which is where you're going, and motor response variables. Those are our X variables. And our Y variables are brain recordings from, say, 100,000 points in the brain. This is a 3 Tesla MRI machine, records about 100,000 points. The new MRI machine we're building here at Berkeley will get 100 times more data. We'll get 1 million points. It'll be really cool. So that's basically our data set. There's a bunch of X's and there's a bunch of Y's. Yes?
What is the frequency for temporal resolution of it?
Oh, you don't want to know. So MRI, I should just mention, MRI is the greatest non-invasive imaging method ever developed. It just beats every other method like EEG and anything else to heck. It still sucks. The resolution of MRI, the temporal resolution, is about a second. There's almost no information in the MRI signals at a time scale faster than 1 second. And that's because MRI measures changes in metabolic activity that are reflected in oxygen flow in the blood vessels. And the coupling between neural activity, neuron can fire about 100 times a second, and blood changes in blood flow, blood oxygenation is very, very slow. Yes?
Can you make the town busier?
Yes, this is just happens to be a Sunday morning. So yeah, there are other conditions where there's
And pedestrians crossing all
and
that.
pedestrians crossing. Yeah.
I presume you'll tell us later, but presumably you have more than one subject. And how consistent is this? Is the story from subject?
I'll show you in just a second, but all the workflow in our lab is again, I don't do anything like anybody else in neuroscience does it. We never do things at the group level. What we do is we do the whole experiment in each individual subject.
I didn't mean at the group level, but the same experience.
Yeah, I'll show you in a second. So, okay, so we got some X variables, we got some Y variables. That means we got a regression problem. And you know, most neuroscience problems end up being regression problems. So, okay, fine, we're gonna do a regression problem.
When you say regression, you mean linear regression?
Well, that's the question. What kind of regression do we want to use, right?
Okay.
Some form of regression. Okay.
Supervised learning.
So what?
Supervised learning.
Well, yeah. I mean, it could be a network, right? So that's the issue. Is what should we do? It's going to be a regression problem of some form, some very general form. We got some X's. We want to predict some Y's, right? So traditionally, the way you do this is basically you just use, you know, your standard regression framework for doing this. You interpret it as Bayesians, but you implement it just as a standard GLM. Now, if you do a linear model, the brain's highly nonlinear, so this would be kind of dumb and wouldn't get you very far. So generally what we do in my lab, I would say 95% of the work is this linearized model, which is basically we take the stimulus, task, and motor variables, and we shove them through some complicated computational model that is meant to produce an intermediate representation that we think might exist somewhere in the brain. And then we regress that representation onto the brain data. And as long as everything's stationary, that should always work because you can always stick a linearity at the end of any arbitrary nonlinear system and it's the same system, right? If the brain's non-stationary, you're screwed, but let's not think about that because we're all screwed all the time. That's non-stationarity is bad. Okay, so generally we do this and I'll explain how we do this in a minute. Of course, like I said, all the kids want to do this. Let's just stick a neural network in here. And even if you want to do this, stick a neural network in here, there are multiple ways to do it that all have all their own advantages and disadvantages. Generally, this is our linearized regression framework. Essentially, we have usually multiple feature spaces that we're testing simultaneously. In this driving experiment, we actually have 33 different kinds of feature spaces, 33 different models. We want to fit them all simultaneously. They all have their own signal to noise. They all have different signal to noise in different brain regions. So we use essentially a banded ridge regression model that allows us to estimate different regularizers for each of these different feature spaces, and they all get fit simultaneously. So this kind of gives you an idea. We got our stimulus and task, we got our brain activity, we've got some linearizing feature space that we're going to interpose between these two because they're too nonlinear to each other. We can pick a variety of feature spaces. And then we're going to have some training data, some testing data, some trained data, some trained features. We're going to get some model weights, and then we're going to cross-validate on a held out data set.
Are you doing single voxel or are you doing joint modeling?
In this stuff I'll be talking about, we're doing one voxel at a time. I'm a strong believer in starting at the finest grain first and then working your way up. I don't like averaging anything until I absolutely have to. So this is all single voxel. I really hate averaging. Optimal averaging is great. All other averaging sucks, and I hate throwing away information, so I don't like to average without knowing what I'm doing. Okay, and then we're gonna test basically on a variety of statistical tests, because if we don't do significance testing, the psychologist will complain. Generally, we produce a variety of metrics in these experiments. Yeah, we do statistical significance. We don't really care about that. What we really care about is estimating the noise ceiling, and then what fraction of the noise ceiling we're performing. In other words, how good is our prediction performance? So now, where do you get these features? Yes.
Are the features labeled in advance?
So they don't have to be, right? You can, the features can be whatever you want, and you can label them post hoc. So, and because you're doing something post hoc, you might worry, well, wait a minute, you're just going to overfit. So in our lab, not only do we have separate fit and test data sets to prevent overfitting, and then within the fit set, we break that up into separate train and test sets for exploratory data analysis. We also collect separate fit and test subjects. So we'll like say collect 3 subjects, get all the models finalized. Okay, this is it. We're freezing everything, and then we'll just fit in the other 3 subjects to make sure we get the same results. Yeah.
So the input to this model, is it a single still image?
No, the input to the model is the video data.
Okay, so you're feeding in like, you're trying to like how I guess
It'll be
yeah, like how much context is there to the model?
As much as the individual feature spaces need.
Okay.
Like you might have a model that says, I think there's going to be somewhere in the brain that represents where you started from. Throughout the entire trip, it knows where you started from, and that representation is going to be active. That's a theory I have. So I can set a feature space that is just basically where you started from, and it will integrate over all time, or it will be tested at all time points, right? Feature space could be anything. And of course, there's going to be a bunch of feature space that have to do with prospective planning about where you're going to drive, and a bunch of feature space is going to be involved with retrospection and memory about where you were in the past, right? Okay, so now we need to get some features. Where do we get the features from? Well, we can get first off, you can just guess what the features might be. Like probably you know where the buildings are and the stop signs and the people that are wandering around there, and you can get them from the video game data. Because I have the subject view, I can do a semantic segmentation. I know where you're fixating, so I know what you're attending to. I can get all kinds of features. This would just be semantic features from this, right? But I also have optical flow, I have color, and I have all the behavioral data. And so this actually just shows the results of one feature space, which is semantic segmentation. This game happens to give you 16 categories of features, and you can color code them according to PCA of the features, and then kind of plot on a flat map. And this is how we interpret things. This is basically our final data is always feature spaces projected onto the cerebral cortex. And we flatten it out because we want to see the whole thing. And this uses software developed in my lab. All right, so this is a really, like, I'm just glossing over so much information here, so don't worry about it. This is 33 models that we fit. You can see there's path, distance remaining, how far you have to go, your future path you're going to take, the beeline distance remaining, the controls you used, day semantics, affordances, which is where can you drive, where can't you drive, motion energy, spatial semantics. There's all kinds of stuff going on here. Everything we could think of based on the literature that might be represented in the brain during navigation. This comes from huge amounts of animal and human literature. We fit all these models simultaneously, all at once in one subject. And now, of course, you got too much data because altogether, there's 33 feature spaces here, but the number of features here is about somewhere probably around 40,000 features. And we're fitting that to each of 100,000 voxels. So it's a big data fitting problem. We use coronal ridge regression for this because the time axis is very, very short. So coronal ridge regression enables us to solve this fitting problem, even though, you shouldn't be able to. And then we get all these data. We have too much data to think about, so we have to do dimensional reduction. Everybody does the same thing when you do dimensional reduction. You either do PCA or cluster analysis or something, right? Those are just two sides of the same coin. And so you get about 5 different networks when you do this. This happens to be a TSNE, which I'm not a big fan of, but that's what's shown here. You get future navigation, motor, visual inputs, which is what you see, and past navigation.
Not sensitive to the type of dimensionality reduction that you're using. I mean,
It is. I'm just showing you one example here.
okay.
Obviously, if we do PCA, we're going to get a different result than TSNE
Yeah.
or non-negative matrix factorization, whatever the heck you want to do, right? And so generally, we do multiple things. There's no right thing to do. So I was taught when I was a graduate student by my very wise old advisor, if there's no right thing to do it as many wrong ways as possible and see if the results you get are consistent. And that's generally my philosophy. So this just kind of shows you, the data are rich and high dimensional. You can also do PCA. So if you do PCA on these data, which is just a different method of dimensional direction, you get kind of 3 functional networks: a motor network, a visual network, and a navigation network. Okay, you should all at this point go, duh, because if I asked you beforehand, before I showed you this data, what would the 3 subdivisions be? That's probably what you pick. But now the cool thing is we can kind of see where they're located across the cerebral cortex. So this is 3 subjects. These networks are fairly consistent with each other. Yes.
How do you know the first one is motor and not the second or the third?
What you do is after you get the PC, you have to look and see what it's loading on. Right. So it's loading on the controls. Also, this happens to be where your motor system is located, and this is where your visual system is located in the human brain.
How did you map the original data into the PC?
I just want to mention this was only the first 5 minutes of the talk. So go ahead. What?
Yeah.
The night is.
Go ahead.
No. Continue on, as long as you.
Yeah, we have an hour afterwards. So
Yeah, it's fine with me. I'm a professional. I'll talk till tomorrow. But anyway.
So how do you know which area to bring your first PC map to?
Well, again, you take the principal components, and so each voxel has a projection into the PCs. So now, if you just take the first 3 PCs, you can assign them to the red, green, and blue gun, and then you can assign that voxel the color of its projection into this first 3 PC space. It's just a quick and dirty way to try to see where things lie.
But PC is on the input, the X. That's a part of it.
Well, the PC is a dimensional reduction on a matrix that's voxels by feature weights.
Oh, the feature you already
Right.
have the voxel.
It's a regression matrix that we're PCing.
You have the voxel in there already.
Yeah, voxels by weights. So this is 100,000 by 50,000, say, matrix.
I thought you don't have the voxel. Okay.
And we get one of those matrices for each subject, right? So usually when you get these PCs, what you do is you stack the matrices all together, so you're getting PCs for all the subjects, and you don't have to do that. It's a handy thing to do. All right, these subjects are pretty consistent. Now, what neuroscientists really want to know is which model is important and which model is not important. And in the regression framework, that ends up being basically a variance partitioning game that you have to play. And we all know that variance partitioning works great if you have 3 variables and then it all breaks down and goes to hell. Don't worry about that. Please don't ask me any questions. Pretend that there's no real weird wizard behind the curtain and just go with it. This is kind of the variance partitioning scheme for these different variables. You can see that some things like affordances, where can you drive? Future path, where am I going to go in the future? Steering and controls. Well, this is other models. That's not it. Motion energy, which is low-level visual features. Gaze semantics, which is what is the semantic category of the thing you're looking at. These are most of the variables that account for most of the variance. And sadly, all the variables that people who do navigation research actually care about, which are these very subtle kinds of navigation-related feature spaces, they account for a vanishingly small fraction of the variance. And I think that's just, those variables are really important, but they're only important for a subset of brain areas. Some of those brain areas, like the hippocampus, tend to be very far away from the antennas in our MRI machine, and therefore have low signal to noise, so they don't end up contributing very much.
Are these directions orthogonal? They're not exactly orthogonal.
These models are not orthogonal at all. That's why variance partitioning is kind of it's a fraught exercise with any complicated data set like this. But you got to do something, right? So, it's kind of funny. This meeting is about interpreting deep learning models. But if you think about this, fitting 33 feature spaces, just using the GLM, we don't even know how to interpret those data. So, and that's a 200-year-old method, right? So kind of scary. I should also mention that, as I said, these brain representations are not static, they're dynamic. If you go home tonight and your cat Fluffy is all of a sudden missing, your brain tries to become a giant Fluffy detector. Where is Fluffy? Where is Fluffy? Every place in your brain tries as hard as it can to be optimized for the Fluffy detection task. And brain areas like near your visual system, your primary visual cortex, they don't change very much. They're just going to keep signaling the outside world. But your prefrontal cortex, every neuron will get sucked into this Fluffy detection enterprise and will become a giant Fluffy detector. And that's just how the brain works. It's this dynamic network that reconfigures its representation based on the task. And you can see that here. When you're in this driving task, this is basically a neutral driving state versus a task-directed driving state. A bunch of representations shift towards poles, which are where the traffic signals are, sidewalks, and other vehicles, and traffic signs, which are all the things you need to pay attention to in this task. Okay, so fine. Does that experiment make me happy? No. Why not? Well, okay, it's like the most massive experiment anybody's done on a data set like this, but we're only accounting for like, I don't know, 50% of the variance we have in the data. So there's a bunch of other signal that we should be able to model. And I don't know what those feature spaces are. So as I said, if you're under 25, your first thought is, why don't you use a neural network trained to drive in this environment, and then you'd solve all the problems. So there's a long history of people trying to use neural networks in neuroscience to explain data. In fact, the very first paper ever that used a neural network was a 3-layer neural network in the good old days to directly fit to neural data was from Yang Dan, who's here at UC Berkeley, and her students, Brian Lau and her postdoc here at Stanley. And a few years later, we did the same thing. This is a pretty simple enterprise. You've got some neural data that you collected. You just try to basically train your neural network by backprop. And then the models will fit really well if you have enough data. And then you try to interpret them. And of course, to interpret them, you have to project them into some space that means something, right? And that's fine. You can do this. But this is never going to work with the data sets we have because we're data limited. We never have enough data to fit a full neural network. So although in some ways this might be a great thing to do, it's out of the possibility of things we can do. So that's useless. So what people do in this field, for the most part, is they use a pre-trained neural network, train on a different task, and they take the features from that network, and then they use the GLM to fit them to their data. And that's a lot better because now the network has learned something that might be useful, and you can figure out what in the learned network is useful for you. So in the vision world, where this has been applied, in the vision world, that where this is done, no, that's not what's done usually, because people who are doing neuroscience don't view themselves in the business of training networks, right? So they'll take the networks from another source. So for example, you'll take AlexNet, or you'll take VGG, and you'll use the features trained in those networks, which were all trained on static images, and you'll use those features to fit your data. And those models actually do a pretty good job. The first person who did this recently, the kind of the recent, post 3-layer network world was Jim DiCarlo's group at MIT, and they showed that these models do a good job of predicting neural responses to natural images across a wide range of the visual hierarchy. So this is a perfectly good thing to do. We've also done this. This is a study from a former student that spent some time in my lab. He was Jitendra student. He's now a professor at MIT, Pulkat Agrawal. And he used AlexNet, actually, to fit voxels in the early visual system. So the visual system, these are all the early visual areas, early intermediate visual areas in the early visual hierarchy. And what he found is that different layers of AlexNet fit different locations in the visual hierarchy. I thought this was a boring result because to me, it's obvious the brain is a hierarchical network trained to represent natural images. So it's going to represent the hierarchical statistics of natural images. AlexNet is a hierarchical network trained to represent natural image. So it's going to represent the hierarchical statistics of natural images. The interesting result would be if those things weren't the same, right? The fact they're the same just means they learn the same thing. And that to me doesn't that interesting. But everybody else got very excited about this. There are some problems using the networks in this way. Mainly that the layers of deep networks are very, very highly correlated with one another unless you take care to decorrelate them. And basically nobody tries to do that. So, but what you can do is you can use banded ridge regression to fit the individual layers of neural networks separately. And banded ridge regression will essentially do, it will adjudicate between the different correlated layers, and it will select the appropriate layer for each voxel. And this kind of cleans up your data lot. This is kind of an ongoing line of research in our lab. It's how to use neural networks to get a fairer answer about what is being represented in the brain that's not contaminated by the biases in the neural networks themselves. Okay, but in the last part of the talk, which I will try to make quick, I want to talk about another approach to using neural networks and deep learning that nobody's mentioned yet. And that is using the really great infrastructure for fitting deep networks to fit an architected neural network that is bespoke for your purposes. So I have long, for like the last 40 years, studied this visual area called area V4. It's in the middle of the visual hierarchy. In the old days, you know, when we had poorer methods, you can only study one area at a time. Couldn't study the whole brain. So I started studying V4. And there's no good model of V4. It's a really important intermediate visual area. There's no computational model of V4 that does any good at all. So we tried to solve that problem. And we tried to solve it by recording from single V4 neurons for weeks and months. So by doing that, this gave us a data set that was far, far larger than anybody else's data set. So rather than having, say, 10 minutes of stimulus response histograms for a neuron, we would have, say, 4 hours of data from a single neuron. And over several years, we collected data from 300 neurons in 2 animals. And that was a data set's 100 times bigger than any other data set out there for this area. So then we said, okay, now we want to fit neural networks. We still don't have enough data to just fit a general neural network. And plus, then we would have an interpretation problem. So instead, let's try to fit an architected neural network that's designed to reflect mechanisms, computational mechanisms in the visual system that we know exist. So that turns out to be this thing here. This is a cascade. We begin by taking our stimuli and doing a log polar transform on them, and that's because your primary visual cortex does a log polar transform on the retinal image to emphasize the phobia. Then we already know we have really good models in neuroscience for primary visual cortex. This is an area that's been studied for 60 years. There are really good models for this. They're used all the time in computer vision. It's the motion energy model. So V1, we implement a motion energy model using neural network architecture, deep network architecture. Then we take this V1 output and rectify it and shove it through a scattering transform, essentially mixing model, and shove it into another V1. So this V2 module is essentially exactly the same as this V1 module. It's another set of motion energy filters that apply to the output of V1. Then there's another scattering transform, which is just mixing the different inputs. And then that goes into essentially another V1 model. So the cool thing about this model is it's kind of a hypothesis that as you develop through this visual hierarchy, each level of visual hierarchy is essentially performing the same operation as the earlier level. It's just doing it on the rectified and remixed output. Yes?
Mixing step there's no like convolutional layering, or it's like all like individual like pixelized like one by one.
Don't even go there. There's a lot of weird local convolutions that go on here because you have to deal with color and time and space, but they're kind of lost in the details on this level. So this is locally convolutional. It has to preserve space, obviously, right? So
Why 4 model? This looks like some kind of an arbitrary decision. Why not 3? Why not 10?
Oh, well, so it's an arbitrary decision if you're not like, a neurophysiology insider, right? So this model for V1 is like the canonical model for V1. It's like the model everybody believes. So then we have 2 areas from V1. We have V1 mostly projects in primates and humans to V2, and V2 mostly projects to V4. So down the ventral stream, which is the pattern recognition stream of your brain. So these 3 layers, that's like set by the anatomy. And then the only question is, what do we want to do in V2 and V4? And since nobody has any idea, the easiest thing is, well, let's just assume that what happened when you, as a primate, developed a new visual area is essentially just did the same thing and rerouted the inputs. So it looks arbitrary but I would, rather than arbitrary, I would replace it with the word elegant, just because it sounds better. All right. And then we compared that to a VGG model. And the really cool thing about this is you notice the number of parameters in this model is 20,000. If you take a pre-trained VGG model and try to fit to these data, you're talking about, millions of parameters, right? So this model is way lower dimensional than taking pre-trained features from VGG. Way, way, way lower dimensional. And it's anatomically plausible. Every single thing in this model, we know how to build out of neurons, out of local circuit elements in neurons. VGG, we have no idea how to build that. So that's kind of cool. I really like that for this reason. All right, and to make a long story short, since we're running out of time, this thing predicts really well. This is VGG versus our model, and you can see our model basically consistently gives you better predictions. This prediction quality for these neurons is about the same as the predictions we always used to get from primary visual cortex. So since everybody believes we have a good model of primary visual cortex, by the criterion of prediction accuracy, we now have a good model of V4. The one thing I did want to mention here is this plot. This plot shows the prediction performance versus the amount of data we have. And if we had saturated, we would see flat curves out here where we're not improving prediction anymore. You notice none of these curves, maybe this red one, but this one neuron, this is 4 different neurons. None of these curves are flat, meaning we don't have enough data. We're already collecting 10 to the 6th frames, 100 times more data than anybody else has, and we still haven't saturated our ability to fit this model, even this simple HCE model. So this, again, I keep repeating myself, neuroscience is data limited. Even when you do everything you can to collect infinitely large data sets that no other human would collect, you're still data limited. All right, now we have a problem, which is that this model is like all these stages are nonlinear. It's like hideous to try to interpret what's going on here. There's no easy way to deal with this and try to interpret it. We can interpret the anatomy. We know what computation is being performed, but I can't go home and tell my wife, oh, here's what V4 does, right? Which is what everybody wants to know. Is it curvature? Is it texture? Is it color? What is V4 doing, right? Everybody wants a story. And I can't give you a story based on this model. So the way we dealt with this was we actually took a trick that we learned from Bin Yu, and we basically determined through an iterative process the optimal pattern that would drive all the V4 neurons in our sample. And you can see these optimal patterns here on these gray. I should mention that these are spatio-temporal patterns, right? Because we're building a full space-time model, but I'm just marginalizing over time for these plots. And so on the left, you can see the V4 receptive fields, which is just the pattern that drives the V4 neuron. On the right is the video frame that elicited the largest response from the neuron in the experiment. And in the middle, they overlap. And some of these are pretty remarkable. Like this is a prairie pheasant. And you can see that this prairie pheasant perfectly matches this weird leaf-shaped V4 receptive field. And it was the stimulus that drove the cell the highest. So you can use this kind of method to try to interpret the V4 cells. But if you look at these receptive fields, they're hideous. No, you can't look at them and figure out what's going on. So this didn't really work without further analysis. So to do that, you got to do dimensionality reduction. Take all 300 neurons and you project them into, I'm not going to go over this in detail. It's good to say what we did. You take your neurons, you project them into some subspace that's appropriate. Like you can take the spatial embedding or a color embedding. You can take a global spatial embedding or a local one, whatever you want. And then once you get all the neurons in this embedding, then you can just do the normal things everybody does, which is either PCA or cluster analysis or something like that. And when we did this, we found that basically we could account for every report of selectivity in V4 that anybody's ever made over the past 40 years. Okay, so that's about it. So I already went long, so I'm not going to belabor the point. Just want to say, neuroscience is data limited. That severely limits the neural network and machine learning applications we can use in neuroscience. All the machine learning stuff that relates to regression still works. The deep network stuff doesn't work so well, but there are ways around it. That doesn't solve any of the interpretation problems, though. The brain is complicated, and interpreting the brain is also complicated. Thanks.
Before we take a pause to set up for the panel, any questions? Quick questions? Yeah.
I have a thanks for the very nice talk. I have a very quick question about the receptive field analysis. Is it fair to assume that there is only one optimum that is relevant?
Good question. I didn't have that slide in here. So when you're estimating these predicted optimal patterns, you don't know that that's a convex optimization problem. There might be multiple optimal patterns that are required. So we actually, for each neuron, we regenerated the optimal patterns either 100 or 1,000 times, I can't remember which. And we looked at the scatter for them. And what we found is most neurons have very compact optima, which actually kind of surprised me. A few neurons have 2 or 3 optima, but the question isn't whether a neuron has a very compact optimum or a few scatter points. The question is, how close together are the predicted optima for one neuron versus another neuron? And in that case, they're all very far apart. So even when a neuron is a little less compact than you might like, it is still very, very different from all of the other V4 neurons.
Do you think that that's the case, especially for V4, or is that a finding that you find in
I'm going to say that's a finding that's general. Okay. 20 years ago now, we showed that even V1 neurons, for those of you who are vision people, V1 is primary visual cortex is as dumb as a bag of rocks. And the standard model of primary visual cortex is a motion energy filter. And motion energy filters, they're all going to be very highly correlated with each other.
But wouldn't you expect that higher brain regions would have more optima?
Oh, hang on a sec. So, yes, but not in the way you think. Hang on a sec. So in V1, if you consider what is known as the non-classical surround, which is a region around the motion energy filter that modulates its responses, and you look at how that non-classical complicated modulatory response affects the neuron's response profile in natural vision, what it does is it decorrelates all the motion energy filters. So V1 neurons are also very, very decorrelated with each other because of this non-classical surround. As you progress farther up the visual processing stream, the distinction between the classical and non-classical surround kind of disappears, and neurons just become more complicated. And they still retain this property of being very, very different from one another. Now, your question is, well, if I'm in prefrontal cortex, say, someplace really spooky where nobody has any idea what's happening up there, are those neurons, do they have local compact optima or distributed optima? And my response would be, they have distributed optima because of attention. So attention and learning have an effect that is directly proportional to the distance of the layer of the neural network from the motor and perceptual surfaces. So if you think about your eyeball, those neurons are just always doing the same thing. If you look at V1, which is only 2 synapses up from the eyeball, those neurons are almost always doing the same thing. If you look at V4, which is the area that I described today, the attention has about a 15% effect on neurons' responses. If you look in the tip of the anterior pole, those neurons are completely modulated by attention. They can be completely switched on and off. In prefrontal cortex, a dog neuron becomes a cat neuron or a learning to drive neuron or anything. The prefrontal cortex is very, very, very flexible. So if you think about attention states and learning and top-down control, then these neurons have many, many, many optima.
Is it the same thing as plasticity, or attention modulate plasticity? Plasticity can also happen for other reasons?
Well, I have opinions about this, but since this isn't my field, you just take it with a grain of salt. My personal feeling is that attention is essentially just the shortest term version of plasticity, right? That is sort of top down under your control. And basically learning is just the time integral of attention. If you attend to something a lot over time, that becomes learning. The temporary changes in the network become more permanent because of repetition. And there's a lot of evidence to suggest that's probably true.
Thanks. Thanks a lot, Jack. So if you want, get up, walk around for 2 or 3 minutes at most.